Hi learners. So today we will talk about the advances in ELISA. This is ELISA series are going on, and we have talked about the normal ELISA, in which we have discussed about the principle, then we have talked about direct indirect ELISA, then competitive and sandwich ELISA. So today is part 4 of ELISA, and advances in ELISA we will be discussing. So there is multiple and the portable ELISA. So it's a new technique used in solid phase made up of an immune sorbent polystyrene rod with 8 to 12 protruding ogive pins. So here is a pin, whereas in normal ELISA we have that wells. So these ogives held in a special frame which is suitable for moving 8 to 12 multi-catcher devices, and it simultaneously immerse the pin in the microwell containing the ELISA reagents. So pins are sensitized with the different catchers, and different tests are carried out in one shot. So no lab equipment are needed for it. So distributing the sample, the multi-catcher device is entirely immersed and incubated in the tube where the sample was collected. Then followed by the washing step. So washing can be made using the micro shower dispenser, tap water or pre-filled microplate. Then distributing conjugate and chromogenic substrate. So pre-filled 96 well plates or the microstrips with the needed reagents are used and sealed and stored until their use. So the kits are for 8 or 12 samples. It contains one microplate which is pre-filled with the conjugate and one microplate pre-filled with the chromogenic substrate. Now kit for the single sample, it only contains the one microplate which is pre-filled with the washing solution, conjugate and the chromogenic substrate in the different columns. Sample enrichment and the sample concentrations are not needed for this multiple or portable ogives, and the test sample aliquot increases, so it will ease the highly diluted target detection. So the system and the component uses. So this is the 8 or 12 pin COM like ogives, and there is a rack for it, and there is a 96 well plate with 12 into 8 pin COMs or 8 into 12 pin COMs. So the entire device is immersed in the test tube containing the collected sample, and the following steps like washing, incubation in the conjugate and incubation in the chromogenous are carried out by dipping the ogives in the microwell of the standard microplate are pre-filled with the reagents. So here there is a dip in the multi-catcher device in each collected sample and incubation. Then we will lift the device and wash it. Then assemble the device in the frame and dip the pin in the conjugate contained in a pre-filled microplate and incubate it again. Then we will lift the frame and do the washing. Then dip the pins in the chromogenic substrate. There we will be adding in normal ELISA. Here we are dipping the pins. So we will dip the pin in the chromogenic substrate contained in the pre-filled microplate and incubate it, and reading can be performed virtually or by using the ELISA readers. Now the ELISPOT. So ELISPOT is a sensitive technique for detection of the cytokines released by the immune cells. So the method closely with the same as the ELISA technique. So it is developed for the detection of individual B cells secreting antigen specific antibodies, and adapted for the detection of individual cells secreting specific cytokine or other antigen. So like ELISA, the bound cytokine can be detected by the secondary antibody coupled with the detection system. So unlike ELISA, the detected analyte is visualized as color spot, with each spot representing an individual analyte secreting the cells. Now these spots are usually counted with an automated ELISpot reader to capture the microwell images and analyze the spot number and the size. So here is the representation of the ELISpot. So the antigen stimulated cells are transferred onto pre-coated prepare plates, which is biotinylated anti-cytokine antibody is added to it. Then the developing reagent is added and the spot formation, that is cytokine secretion, can be seen. Advantages of ELISpot: that it is sensitive than ELISA, allowing the detection of cytokine release at lower concentration or by the small number of cells. It is versatile. That means it is easily tailored to detect a wide variety of specific secretion, easy to perform and quantify the result, provide evidence of the secretory product of individual activated or responding cells. The applications of ELISPOT. So it can be used for vaccine development, cancer research, allergy, transplantation research, monocyte macrophages, dendritic cell characterization, apolipoprotein analysis, and for the veterinary research. Thank you. Like, share, and subscribe the video, guys.